So thank you everyone for coming. This summer I spent my time learning about climate models and how we use them to model phytoplankton in our oceans. So first off, I want to talk about why we model phytoplankton. Well, they're the base of the oceanic food chain. They are present in the lighted regions of all the world's oceans, and they're responsible for between 50 and 70% of our atmosphere's oxygen. They're also very sensitive to climate change. So if you see community changes in phytoplankton occurring in our oceans, then we can also tell that our oceans are changing with climate change. And different groups of phytoplankton occur in different areas of the world, and that's referred to as phytoplankton biogeography. So for this project, we focused on two main groups of phytoplankton. The first of which is diatoms, which are larger phytoplankton that prefer subpolar, colder waters with a lot of nutrients. And our second group is small phytoplankton, which due to their smaller surface area to volume ratio, they can take in nutrients faster than diatoms, and they can also live in more nutrient-poor waters that diatoms aren't able to grow in. Our study area for this project is the Southern Ocean, which is the ocean around Antarctica. And we chose this location because it is experiencing a lot of change along with climate change. So how do we model phytoplankton? We use Earth system models, specifically the community Earth system model. And these models are capable of simulating atmospheric, sea ice, ocean, and land surface processes, with biosphere and biogeochemistry recently being added. And within biogeochemistry is phytoplankton. So what these models do is they separate the Earth into a grid system, with the horizontal grid being based off of latitude and longitude, and the vertical grid being based off of height and pressure, and this grid extends up into the atmosphere and down into the ocean. And the grid cell sizes for different models can vary. So in other words, model resolution can vary. And the purpose of these Earth system models is to eventually be able to accurately predict the future. So this is a figure from Moore et al. In 2004. And on your top panel is a model of phytoplankton primary production. And on your bottom panel is a real satellite estimate visual showing real-world data of phytoplankton primary production. And so the agreement between these two panels shows that models are sufficiently capturing ecological complexity at the global scale. And as we progress in our supercomputers and our models, the ultimate goal is to be able to find out what's going to happen with our planet in the future. So the question for this project is, how does phytoplankton biogeography in the Southern Ocean vary across fine and coarse model resolutions? So we were provided model output in coarse and fine resolution from the National Center for Atmospheric Research. And these are StereoNet projections of the Southern Ocean. And I labeled major land masses that you would see. So Antarctica is in the middle, and the southern portions of South America, New Zealand, and Australia are also visible. I also labeled some oceanic locations. So you have the Weddell Sea, the Ross Sea, and the Kerguelen Islands. And so the coarse resolution is a 49-year run with forced real-world data. So what they did was they took historical atmospheric measurements and they forced those measurements onto an oceanic model. And this grid is a 1 by 1 degree grid. And in the fine resolution, it's a 5-year run with today's climate being simulated, along with today's ocean being simulated. So essentially, our oceans and our atmospheres are communicating with each other. And this grid is at 0.1 by 0.1 degrees. And so what I want you to see here is these StereoNet projections of sea surface temperature in degrees Celsius. So the coarse resolution, it's a little more muddled between the differences in sea surface temperature. And in the high resolution, you get a lot more definition between the temperatures. And I want to point out up in the Patagonian shelf area on the side of South America, you see this beautiful filament of colder water going up the Patagonian shelf that you didn't see in the fine resolution, that you do not see in the coarse resolution. So some pros and cons of our resolutions. In the coarse resolution, it takes a lot less to compute, and it's easier to analyze our entire planet, but you get less detail, and it's only oceanic processes. So if you're going to spend your time and your money, you got to think about what resolution you want to run this at. And this is what our models are run on, so NCAR's Yellowstone supercomputer up in Wyoming. And so pros and cons of the fine resolution, you get a lot more detail, and also it's oceanic and atmospheric processes interacting with each other, just as the real world does. But it's exponentially more in computational cost to run a finer resolution, and it's more difficult to model the entire planet. So if you have a coarse resolution and you want to run it at just 2 times as fine, it takes 10 times as long to compute that. So our methods involved looking at 5 factors over our fine and coarse resolution models, and that includes total biomass, biogeography, ice cover, sea surface temperature, and dissolved inorganic iron, which is a very important nutrient for phytoplankton growth. So our first result is a bar graph of integrated phytoplankton carbon at coarse and fine resolution. So on your x-axis is resolution, coarse being blue, fine being green, and on your y-axis is petagrams of carbon, which is a proxy for phytoplankton biomass. And this is the sum of diatoms and small phytoplankton for both resolutions. And we found that there was a 12.5% difference in phytoplankton biomass being simulated at the fine resolution, more so than at the coarse resolution. So then we wanted to see how this varied over our phytoplankton groups. So we split up these resolutions into the 2 respective groups, with your x-axis, the phytoplankton group, so diatoms on your left and small phytoplankton on your right, and again on your x-axis, petagrams of carbon. And we found that the coarse resolution was simulating 28.6% more diatoms than the fine resolution, and that the fine resolution was simulating 36.4% more small phytoplankton than diatoms. So then we wanted to be able to visually represent how our phytoplankton were varying over the Southern Ocean. So what we did was we made stereographic projection maps of our fine resolution, and we set that to the coarse resolution, and we averaged all of these measurements in order to facilitate comparison of a 49-year run and a 5-year run. And then we subtracted the coarse resolution from that to get the difference. And what you see here, the middle figure is the difference between the fine and coarse resolution. So this is average diatom carbon in millimoles per cubic meter in the surface of the ocean. And if it's blue, that means that more diatoms are being simulated at the coarse resolution. And if it's red, then more diatoms are being simulated at the fine resolution. So we also did this for average small phytoplankton carbon. And I want to point out in the Patagonian shelf that in the fine resolution, diatoms are occurring around this smaller island and up this filament. And also in the fine resolution, small phytoplankton are occurring all along the Patagonian shelf and not where the diatoms occur. So this is a beautiful example of phytoplankton biogeography where they'd fit like puzzle pieces as diatoms are growing where phytoplankton aren't. So we also looked at fractional ice cover and sea surface temperature. And I want to point out this very major difference in the Weddell Sea. So in the coarse resolution, you're seeing a very large swath of ice being simulated, while not very much ice being simulated in the fine resolution. So this matches with sea surface temperatures. So in the fine resolution, you're getting higher temperatures in the Weddell Sea and lower temperatures in the coarse resolution. So it makes sense that the fine resolution is simulating less ice if it's warmer. We also looked at dissolved inorganic iron, and again, the Patagonian shelf really sticks out as a difference across resolutions with this filament of ironless water up the Patagonian shelf being visible in the fine resolution in figure E3 and not visible as much in figure E1 at the coarse resolution. So these are our figures of average small phytoplankton and diatom difference across resolution. And here you can really see that small phytoplankton, a large amount of red being shown, so they're being more so simulated in the fine resolution, and a lot of blue, so more diatoms being simulated at the coarse resolution. So a few takeaways for this project. We found that diatoms dominated in the coarse resolution, while in the fine resolution, small phytoplankton dominated. We also found that at the coarse resolution, you had lower sea surface temperature and more ice cover, while at the fine resolution, you had higher sea surface temperatures and less ice cover. And this makes sense because diatoms, they like that subpolar water, those nutrients. And if you have a warming sea, then that means that diatoms aren't going to be able to survive in those higher temperatures. And that means that smaller phytoplankton are going to be able to take what nutrients are available, and they're going to take over where diatoms used to be able to occur. So our final takeaway is when simulating phytoplankton biogeography, we recommend you use a fine resolution as it just seems more capable of picking out those details of phytoplankton community structure and defining even like how phytoplankton interact with currents and whatnot. And the coarse resolution just seems a little too broad in grid cell size to really capture those details. So future work for this project would involve comparing our model output to real-world satellite estimates. And we could look at phytoplankton biomass, as these two figures on the left show, or phytoplankton mean chlorophyll at the top right corner. And mean chlorophyll is a chlorophyll, so pigment that phytoplankton produce as their photosynthesizers. And that's another proxy for finding out where phytoplankton are occurring in the ocean. So I would just like to thank everyone at UNAVCO for supporting my coming for this internship. It was wonderful, and everyone else who was so important for helping me through this project. My mentor, Nicole Lewandowski, she was wonderful, and I made a lot of great friends. And I'd also like to thank the National Science Foundation for making this experience possible. So does anyone have any questions? Thank you.
Lots of questions. That was an awesome talk, so thank you. That was great. I was wondering, why exactly is there a difference in sea surface temperature or ice predicted in the fine versus the coarse model?
So in the coarse model, you have atmospheric data being taken from the past, so 1958 to 2007. And then the fine model, you have today's atmosphere. So you have that gradual warming from 49 years ago to today. So that's why there's such a difference in ice occurrence and sea surface temperature.
That makes sense. So you think that the fine resolution atmosphere is more similar to what we're seeing today?
Yes, it's based off of today's climate.
Right. So that's why you think it's actually predicting what we're observing.
Yes.
Yeah. So okay. Cool. Thanks.
Thanks, Kaylee.
Would it be possible to develop some sort of, I don't know, let's call it margin of error then, so that using this research so that you could utilize rough coarse models over a wider area, but be able to sort of apply some sort of error to your findings so that you're able to take advantages of the wider studies?
I believe that would be a wonderful thing. I'm a geology person and not a climate modeling person, so I learned a lot this summer. But I think that would be really beneficial because the amount of time it takes to run a fine resolution model, and especially since it doesn't run the whole globe, at least in a few years to run it, I think that would be certainly a wonderful thing. And I'm sure people at NCAR are working on that or already have that in place.
Helen.
Great talk, thank you. I was kind of following up is, would it be possible to possibly merge the two in a way that you have multi-grid sizes? So places where we know we have a lot of current or a lot of human activity, we could do the finer scale and then build it into a larger global scale?
I don't see why not. Yeah.
Okay. Any other questions? All right. Thanks, Leslie.
Thank you.